This is Michael Smith of MedPage Today. I'm speaking with Dr. Barry Zingman of the Montefiore Medical Center in New York. Dr. Zingman is at the conference on retroviruses and opportunistic infections here in Boston, and he's reporting data on Reassuring, a CCR5 inhibitor that is aimed at blocking the entry of HIV into its target cells. Dr. Zingman, can you give me a description of the study? This is the Victor E1 study. Tell me how it was designed and who was in it.
Yes, Victor E1 is a 48-week phase 2 study that was done in 12 countries around the world. It is a randomized double-blind placebo-controlled study of 2 different doses of Reassuring, 30 milligrams or 20 milligrams versus placebo, in addition to a ritonavir boosted protease inhibitor containing optimized background therapy.
Okay, and the patients in this study were highly treatment experienced, I think.
Yes, patients who were eligible had to have R5 tropism, but also had to have triple-class experience and had to have at least 1 mutation to nucleoside reverse transcriptase inhibitors and 1 primary protease inhibitor mutation.
So this was a very relatively sick population.
Yes, the end result was that the average CD4 count of enrolled patients was right about 200, and about 85% of patients enrolled had fewer than 3 active drugs in their background therapy.
Wow. The drug, the Reassuring, was in 2 doses, essentially against placebo. What did you find?
So the primary efficacy outpoint was the drop in RNA levels, log 10 drop in RNA levels at week 48. We had secondary endpoints in lowering viral loads to less than 400, less than 50, and there was safety and tolerability endpoints as well. The main outcome of the study, the primary efficacy outpoint, showed that both of the primary efficacy endpoint showed that both of the doses lowered RNA levels about 1.75 logs in comparison to the placebo plus OBT that lowered about 0.7 logs. Both Reassuring arms were superior and statistically significant. And as well, the Reassuring arms, but especially the 30 milligram dose, seemed to do better regardless of the number of active drugs in the background therapy, and also for patients who entered the study with high RNA levels, over 100,000, and also for people with lower CD4 counts. So
the higher dose did better in patients that were harder to treat, essentially.
That's right.
The drug was worrisome in its initial presentation at the Toronto AIDS meeting when researchers noted a seeming signal that it was responsible for malignancies. You didn't see that in this study.
Well, I'd say first that those malignancies in an earlier study were reviewed by an expert panel and judged unlikely to be due to the Reassuring. They were felt to be part of the natural history of a very sick population of people with HIV and AIDS who were in that study. So, of course, that issue has been followed very, very carefully. In the study, we found no malignancies during the study.
Excellent. Finally, the issue of tropism. You're aiming this drug at people with a particular form of the virus that uses the CCR5 co-receptor. There are other entry points. Tell me, what did you find with regard to the viral tropism in these patients?
Right, so in the study, patients were screened for CCR tropic virus, and if they had it, they were able to get into the study. They then had follow-up tropism assays done at baseline, at their very first visit, before they started the drug at all, at week 8, week 24, and week 48. What we found was that a percentage of patients in all 3 arms had emergence of DMX4 virus. This was slightly more frequent in the 30 milligram dose, but when it occurred in the 30 milligram dose, it occurred very early. The occurrence actually was at baseline before first dose, or especially by 8 weeks. Occurred very early, suggesting that it was actually present already in patients before they were treated, and it was the potency of the 30 milligram dose that helped it emerge a little bit earlier. It was there already, but it came out under a potent treatment that was suppressing CCR5 virus. It's notable, though, that in the 30 milligram group, not all of the patients, actually only about half, ended up being discontinued from the study with viral loads that were over 400. In the 20 milligram group and the placebo group, there was a closer correlation between development of DMX4 and discontinuation with a viral load over 400. And whether or not this will continue to be a trend, we look forward to the phase 3 studies, the larger studies. These were a small number of patients. There were about 35 to 39 patients per arm. So we look forward to larger studies to see if this finding is confirmed.
So did you see a difference in efficacy based on this tropism switch?
As I mentioned, the patients in the 30 milligram arm overall did very well, and in a small number of patients, it was not consistent that the finding of a DMX4 variant led to treatment failure. Remember also that some of the patients had emergence of the DMX4 even before they got the first dose, and then those patients would have been treated with microviric plus an optimized background therapy. So all of that in combination did seem to work for many people.
Okay. And I guess the final question is what's the clinical take-home message for this drug? Will it be available soon?
So what I can say right now is that microviric seems to be a very potent oral CCR5 receptor antagonist, and it holds a lot of potential for the future treatment options for people with HIV. And so it is right now in larger scale phase 3 studies, and depending upon the results of those studies, efficacy and safety, that will determine how soon the medication might become available.
Not currently available, though, for physicians?
No, it's not. It's in phase 3 studies.
Right. Thank you very much, Dr. Zingman. I'm Michael Smith, MedPage Today.